What's up, Bobcats? In this lecture, we're going to go over the absorption of amino acids and carbohydrates. But before I get started, make sure to hit that like button and subscribe. So the first thing that I want to point out is what I have drawn here on the board and what's the overall goal here. So for carbohydrates and amino acids, both of these substances need to get into the liver because this is where it gets processed. So in order to get it to the liver, it has to get here into the capillaries, and then it uses the hepatic portal vein, which will transport it to the liver. But in order to do that, it has to get here into the enterocyte, so this small intestine cell. From there, it'll then get into the capillaries. So now what we're going to go over is the process of how these different substances get into the cell. Okay, so we're going to start off with the carbohydrates. And the first thing that I want to point out is number one. So number one, this is what's known as the sodium potassium ATPase pump. And so this is found, so it's within the enterocyte, but it's important for carbohydrates as well as the amino acids. So the whole purpose of the sodium potassium ATPase pump, so what we need to do is pump sodium out of the cell and then pump potassium into the cell. And so what this does, it creates what's known as a electrochemical gradient. And this electrochemical gradient, this is what allows these co-transporters. So these carbohydrates, they are using co-transporters. So some of these co-transporters, one of them is what's known as the SGLT. So this stands for the sodium glucose linked transporter. And what the mechanism that it utilizes, it's what's known as secondary active transport. So because there's typically a higher concentration of sodium outside of the cell here, we have to pump it against its concentration gradient. And in order to do that, we need ATP. So by pumping the sodium out, this creates the electrochemical gradient, right? There's a difference in charge, and it allows this co-transporter for the sodium as well as two different types of monomers to get into this enterocyte. So what it transports is glucose as well as galactose. Okay, so this is for SGLT. So the next one is number 3. So number 3, this is what's known as the Glut2 Transporter. So the Glut2 Transporter utilizes facilitated diffusion, meaning that no ATP is required, and we're just using the concentration gradient, because what we're transporting with Glut2 is sucrose. And with sucrose, there is so the concentration of sucrose is going to be higher in here compared to inside the cell. So by using this protein, it allows it to flow into this enterocyte. Okay, so the next thing, so number 4. So number 4 is what's known as the Glut2 Transporter. And this Glut2 Transporter also utilizes facilitated diffusion. And what travels through this channel is all of these, right? Because now we have glucose, we have galactose, we have sucrose. So all of them are here into the cell, and then now we have to get them out in order to get it here into the capillaries. So that's what this is. So we're transporting glucose, galactose, as well as fructose. Okay, so that's going to do it for carbohydrates. So now we're going to move on to amino acids. So I've already mentioned we're establishing the electrochemical gradient using the sodium potassium ATPase. But now how do we get the amino acids in? So one of the first ones that we'll discuss is it's pretty similar to this one over here. So it's sodium dependent amino acid transporter. So we're utilizing the same mechanism. So whenever sodium gets into the cell, we can then transport one of these single amino acids. Okay, well, what about number 3? So number 3, this is a transporter known as Pep T1. So this is Peptide Transporter 1. So Peptide Transporter 1, it typically transports either tripeptides or dipeptides. So tri is 3, di is 2. And the special part about this is that it's a symport, meaning that in order to get this peptide here in, so to get the peptide in, draw it on this side. In order to get the peptide in, we also transport a proton. So we have a proton peptide symport. And once we get this proton in, we also pump some of these protons out. So you also have number 4. So this is what's known as the proton sodium antiport. So once we pump this hydrogen ion in, we can then pump it out, and then what comes inside, this is sodium. Okay, so number 5, so how do we get these different amino acids here into the capillaries? So they have to travel through number 5. So number 5 is just our amino acid transporter. And this transporter is located on what's known as the basolateral side, meaning that it's on the bottom and then it's lateral. Okay, and so the mechanism that's utilized here, this is also facilitated diffusion. One other thing I would write here, so amino acids, right, we're also using cotransporters here. Cotransporters. And this is going to the liver via the hepatic portal vein. Okay, so that's going to do it for this lecture.